Hi Aztecs, welcome to Module 2B, Chapter 3, Cells, Part 2. Remember, we said I think there's 5 or 6 parts to this chapter in terms of our videos. So this video we're going to talk about organelles. You already have a favorite organelle? I kind of like this guy, the mitochondria, because it makes ATP. And ATP is kind of important in terms of currency for the cell to build things. Your new best friend. Excuse me. So in the last video, we talked about the 4 concepts of the cell theory. First concept, cells are the structural units of all living things. That means they're the smallest unit of life that can survive and reproduce on their own. So there's a lot of single cell protozoa out there. There's just one cell. They survive and they can reproduce. The activity of number 2, the activity of an organism depends on the collective activities of its cells. We talked about that in the previous video. For example, the collective activity would include the activities of your heart cells and your lung cells and your liver cells all working together to make you active. And then, according to the principle of complementarity, function complements structure, structure complements function. For example, your skeletal muscle cells, they have a certain structure that allows them to function to contract your muscles. And we'll look at that molecular structure of a muscle cell later in the class. And the continuity of life has a cellular basis. So as life goes on, from like your grandparents to your parents to you to your kids, continuity of life, it's all about cells. We also talked about this the last video about the cytoplasm, and we're reviewing this because the cytoplasm is where the organelles are. So in the cytoplasm you have the cytosol, which is just like the fluid that suspends everything in the cytoplasm. Organelles, that's what we're talking about this video, and then inclusions are also in the cytoplasm. Okay, the nucleus, the control center of the cell. Why is it the control center? You probably know it has DNA. But before we get to the DNA, if we start superficially, so this whole purple ball is the nucleus, and it's cut. You can see you've got a couple different cuts so you can see inside. Superficially, it has this thing called a nuclear envelope. That's this. So this is nuclear envelope. This is all nuclear envelope. This is all nuclear envelope all the way around. Deep to the nuclear envelope, you have chromatin. That's DNA in its unwound form. And then you have this structure called a nucleolus. And then these orange things are nuclear pores. And then outside the nucleus is the rough ER. But what's interesting, doesn't really point out in this cartoon, but check it out. The part of the membrane of the nucleus right here, check it out, how it folds back. And then, woohoo, it becomes the membrane of the ER. So you have this little continuous membrane from the nuclear envelope to the ER membrane. So there's 3 regions in the nucleus. That's the nuclear envelope, the nucleolus, and chromatin. And we said the nucleus contains DNA. That's genetic material. And DNA, we'll talk a lot about that later, but DNA is necessary for cell reproduction, so to make new cells. And the DNA also specifies all the proteins of your body. So the nuclear envelope, it's a double membrane around the nucleus. So see, you can see how it's a double membrane. So here's one part of the membrane, and then it folds back on itself here. There's the other part of the membrane. That is a double membrane for the nuclear envelope. And then these nuclear pores, these gold things or yellow things, we talked about those, allows for exchange of material between the nucleus and the rest of the cell. And within the nucleus is a jelly-like nucleoplasm. It's kind of like the cytoplasm. This is the nucleoplasm. Nucleoli. Nucleoli is plural. Nucleolus is singular. So the nucleus contains one or more, you could have one or more nucleoli. And that's where you make ribosomes, on the nucleolus. And the ribosomes are these little orange dots out here. The ribosomes, that's where you make proteins. And the ribosomes sit on the endoplasmic reticulum, or the ER. And when the scientists first saw this in a microscope, they're like, oh, there's the ER. It looks like it's rough, but the rough part they were seeing were really these ribosomes. So you make the ribosomes on the nucleolus, and then the ribosomes migrate out into the cytoplasm through these nuclear pores, and then they go sit in the ER. The chromatin. Chromatin is composed of DNA and protein. When the cell is not dividing, you'll see chromatin. When the cell is dividing, you see chromosomes. Oh my gosh, what is this? Check this out. I love this chart. So when you say the word metabolism, most people just go, oh, he has a high metabolism, or she has a high metabolism, or they have low metabolism. That means they can't eat very much. But metabolism is really a complicated chemical events. It's complicated chemical events, plural, that's happening in your cells all the time. And these are all the chemical events over here in this giant chart. And then I just kind of circle the middle of the chart here and kind of bring that part out where it says acetyl-CoA. And then, this isn't a chemistry class, so we're going to talk about most of this chemistry, but we will talk about this chemistry of how you take a glucose molecule, break it down, and make ATP. That's metabolism in a nutshell. Okay, more organelles, mitochondria. Here we go. Mitochondrion is singular. There they are. It's the powerhouse of the cell. That's where you make a whole lot of adenosine triphosphate, and you need ATP if you need to build things. Here's a cartoon that's a little more detailed of the mitochondria. So let's check it out, because we're going to need this when we do metabolism. But you have this outer membrane. So this orange part here, this is all cut from the mitochondria. This is all outer membrane. And it's phospholipid bilayer. And then you have this inner membrane inside the mitochondria. So you have an outer membrane and an inner membrane. And then between the outer membrane and the inner membrane, all this stuff is an intermembrane space. That makes sense. So you get this, see how it kind of folds? The inner membrane kind of folds back and forth. Those folds are called cristae. And then you can see inside the inner membrane, you find ribosomes and granules, and living within this thing called a matrix. And then also within the inner membrane of the mitochondria, you have ATP synthase particles, which you don't care about now, but when we talk about glucose metabolism, they'll become really important. Smooth and rough ER. So we already talked about the rough ER. Rough ER has ribosomes on it. That's where you make proteins. And then there's also another part of the ER called the smooth ER. Real smooth. So the ER is endoplasmic reticulum. We said this rough ER is proteins are made there, and that's very complicated. We'll go over how that happens. So for now, just know the rough ER is where proteins are synthesized, and then we'll go over the gory details of protein synthesis later. The smooth ER, however, we'll talk about that in more detail here. This smooth ER, this is where you make hormones that are steroid based. So for example, testosterone and estrogen. If you are a hormone and you are steroid based, you get made in the smooth ER. Some hormones are proteins and they get made on the rough ER. You also do lipid metabolism in the smooth ER. You make cholesterol in the smooth ER. You can break down glycogen into glucose. So glycogen is just a giant chain of glucose monomers. That's how you store glucose in the form of glycogen. And then in the smooth ER, you can also do detoxification of toxins and calcium storage, which will become really important when we talk about the skeletal system. Okay. All right, so let's check this out. Synthesis and export of a protein by the rough ER. So here's the ER, all this blue stuff. Okay, this is all ER. And here's the ER in the bigger picture. All this blue stuff is ER. Endoplasmic reticulum. And then here's the ribosome that we talked about. And it looks like this ribosome's got two pieces to it. And here's this blue ribbon is a piece of messenger RNA. So the messenger RNA came from DNA. So from DNA, you make messenger RNA, and then that messenger RNA gets translated into a protein on the ribosomes. So each one of these purple balls is an amino acid. You string a bunch of amino acids together and you make a protein. And it looks like they stuck in the ER, they stuck a couple green things on it. It's probably carbohydrates stuck on the protein. And anyway, then after your protein is made, the ER will bud off into this thing called a transport vesicle. Proteins inside the transport vesicle. Where is that transport vesicle going to go, you ask? Well, some of you may already know. It's going to go to the Golgi apparatus. So let's take a look at the details. The protein is synthesized on the ribosome. We talked about that. Protein migrates to the rough ER cistern and folds into its functional shape. So a cistern, it's Latin, I think, for like vessel. Anyway, just the whole space here of the ER is the cistern. That's the rough ER cistern space. And so you trick out your protein there, package the protein into a transport vesicle, and then the transport vesicle goes from the rough ER to the Golgi. We'll look at the Golgi on the next slide, but that's this green stuff here. This is the Golgi apparatus. So in this slide, the ribosomes, the rough ER, and the Golgi, they act in sequence. So ribosome, if you're making a protein, ribosome first, ER second, Golgi third. They act in sequence to synthesize and modify proteins for secretion and for integral plasma membrane proteins. What does that mean? So if you are a protein, you got made here. So you got made on the ribosome, the rough ER. Now you're a protein. Now you go to the Golgi. Then the Golgi can either secrete you. So that's in this cartoon down here. So this vesicle with the protein in it will bud off from the Golgi, and you'll secrete these molecules or proteins that you made. Or the Golgi could also, and I don't think it shows it in this cartoon, but one of these could bud off and just stick proteins in the plasma membrane. Integral, and then those are called integral membrane proteins. Okay, so your protein, you actually, to make a protein, starts off in the DNA. DNA makes RNA. RNA comes to a ribosome and gets translated into a protein. Protein goes into the rough, well, it goes into the lumen or the cistern of the endoplasmic reticulum. Then that vesicle buds off and kind of molds or melds with the Golgi, and you get further processing of the protein inside the Golgi. And then the Golgi vesicles can have several different pathways, but in pathway 1, if you were a protein and you got into a Golgi vesicle, you could be a protein that gets secreted. So that's what this cartoon shows. So these are secretory vesicles that pinched off the Golgi apparatus. They're filled with proteins, and then they get secreted by the process of exocytosis to the outside of the cell. The second pathway, the Golgi vesicle containing plasma membrane components like proteins will fuse with the plasma membrane. So here's it doesn't show the components in it, but here's the Golgi vesicle. It kind of fuses because it's phospholipid bilayer. It fuses with the phospholipid bilayer, and then this part here just becomes part of the phospholipid bilayer. And if there's proteins in there, then now you got proteins in your phospholipid bilayer. And then the third pathway that if you were a protein and you were in a vesicle in the Golgi apparatus, the third thing that could happen to you is you could have been filled with digestive enzymes like lysozyme. Lysozyme can be found in this organelle called a lysosome. And then we'll talk about this later, but this lysosome's got a bunch of enzymes in it that can kill bacteria, for example. And then they can destroy bacteria if they get into that cell. Okay. Golgi apparatus modifies and packages proteins arriving from the rough ER via these transport vesicles here. And then Golgi makes secretory vesicles here. Okay, we talked on the last video about the cell having a skeleton, but it's because it's in the cell, it's a cytoskeleton. And here's the organelles that make up the cytoskeleton. So there's these things called microfilaments, and here's a cartoon of them. And they're made of these little balls called actin, so it's an actin subunit. And this 7 nm means this diameter is 7 nanometers. And then this is cool because this is an actual fluorescent stain, so you can get a fluorescent dye that attaches to these microfilaments made of actin, and then you can look at them in a fluorescent microscope and you can see where they are because they're all blue. They fluoresce blue. And it says it's a bat-like network because there's a lot of actin in the cell. The next part of the cytoskeleton is called an intermediate filament, and it's made of these things called fibrous subunits. And you can see it's a little bigger in diameter. It's 10 nanometers. And then so this big pink thing, that's the nucleus. And then all these purple filaments are intermediate filaments, and they stain with a dye that can fluoresce under a certain wavelength, and you see purple. And then the third part of the cytoskeleton are these things called microtubules, and they're made of tubulin subunits. And look at how wide they are. Look at their diameter, 25 nanometers. Again, the nucleus is fluorescing pink, and then the microtubule network is fluorescing yellow. So we have the microfilaments, their subunits are actin. We have the intermediate filaments, their subunits are fibrous. And then we have microtubules, their subunits are tubulin. Okay, and those are the three different types of protein. These are all proteins that make up the skeleton. Okay, so now let's go back to our cartoon. Shows in our cartoon the microtubules here. Here's the microtubules. Remember, the microtubules are made of these tubulin subunits, and this has got quite a diameter. It's like 25 nanometers, so they're pretty wide. So the microtubules are generated in these things called centrioles. So this whole thing is a centriole, and the microtubules are made from the centriole. And the centrioles and the microtubules direct the formation of the mitotic spindle during cell division, otherwise known as mitosis. Oh, and here's the intermediate filaments, so you can kind of see them here. Here's our cartoon of the intermediate filaments. Remember, they're made of fibrous subunits. Okay, cell extensions. So we've seen this cartoon before. So this is a whole cell. This guy's a whole cell here, and it's got the plasma membrane. It kind of makes these finger-like projections. Yeah, that's all plasma membrane just projecting. And these projections of the plasma membrane are called microvilli. The reason some cells have microvilli is so that you can increase the surface area for absorption. Where do you want to absorb stuff? In your intestine. You want to absorb the nutrients, so you have these microvilli. I'll talk all about this in 202 when we talk about the intestine. Not shown in this cartoon are cilia. Cilia move material across the cell surface. They're located in the respiratory system to move the mucus. So you have in your trachea, you have a ciliary escalator that's made of mucus and cilia. So if bacteria get into the mucus in your trachea and then the cilia, their projections of the cell membrane, the cilia beat upwards. It'll move the mucus and the bacteria to your oral cavity, and then you can spit it out or swallow it, whatever you like. And then another surface extension of the cell would be a flagella. Probably everybody's heard that the sperm have flagella and they swim. The only cell in the human body that has a flagella is sperm. And here's our hero again, your new best friend. Study, study, study. See ya.